I'm going to introduce Dr. Kellie Walters. She is here today as a guest of Dr. Ferrarezi. She is assistant professor in controlled environment plant physiology at the University of Tennessee. Her overall goal in the Walters Lab is to leverage the ability to manipulate the growing environment to alter plant physiology, improving production efficiencies and crop quality. So, and I don't know if you want to add to that any, but
awesome.
I'll let you get moving.
Thank you.
Sorry,
for the
there's no
introduction.
clicker.
You're great. You're great.
All right, thanks.
Awesome. Thank you everyone for coming today and for having me here. So yeah, just came down from Tennessee yesterday and your campus is beautiful and it's been great to see the facilities and what all research you guys are doing. So today I'm going to share a little bit about what we've been doing in my lab at the University of Tennessee. I titled this presentation, Controlling the Environment to Improve Yield and Nutrition. So that's kind of like my overall goal, right? So we're trying to leverage our ability to control this environment to achieve our desired outcomes. So, well, now I can't advance at all.
Click somewhere in
You
the,
click on the screen.
yeah, just click somewhere in the screen.
Nope.
Oh, maybe.
Oh, it's that's.
Try it again. Oh, wait, okay.
Did it go? Nope. Oh, it's been doing that.
Oh, here, maybe if I, nope, okay. Well, while you're messing with that, is it okay if I keep talking?
Yes.
So I like to start off with this picture. These two pictures. So these two pictures here are of lettuce seedlings. They were the same.
Okay.
Awesome. Thank you so much for your help. They're both the same cultivar. Oh, perfect. Thank you. You fixed it all. So these are both the same cultivar of lettuce. They were sown on the same day. This picture was taken on, these two pictures were taken on the same day, not the day where they were sown, but the same day. The only difference in these two pictures is the light intensity that these plants were grown under. Now they look very different, right? Super purple. This one doesn't even almost look like it's a purple leaf cultivar, but it is. But I like it. I like it in that it illustrates how we can alter that environment to drastically alter the appearance of crops. We know we can alter the yield of crops, but also some of the phytonutrients and quality aspects of these crops that we're growing. So that's kind of what we're going to talk about today. So first we'll talk a little bit about what is quality. Quality can be a lot of different things, but what are we going to talk about today? My lab does a lot of different work in controlled environment agriculture. Like, how do I fit it into one seminar that actually makes a cohesive story and sense? So we're going to focus on lighting and mainly light intensity, just to narrow it down a little bit more. And then we're going to talk a little bit about consumer preferences, tying together the production factors to some of the phytonutrients and efficiency, and then what consumers think. And then we've been doing some work for NASA, so I'll share that kind of story at the end. But that's what we're going to go over today. So the overall goal, okay, we're trying to improve crop quality, but we need to maintain yield because ultimately we have this production area. We only have a certain amount of space. And then when we sell that crop, we usually sell it based on weight. So we want quality, but we also don't want to sacrifice yield. So what is quality? A lot of different parameters. These are some of the things, well, I'm not going to talk about all of them today, but flavor. Does something actually taste good? When we're eating it, does it taste like super bitter? If we're growing a culinary herb, does it have that characteristic flavor that you're looking for, or is it way too pungent? Are there markets for both? Nutritional quality. So some of the different beneficial compounds for human health, some of the vitamins, etc. As we alter those growing environments, we can alter the nutritional quality of crops. Post-harvest longevity, we harvest that plant. One of the advantages of controlled environment agriculture is that we can grow our plants wherever. improve that geographical diversification. So not all our lettuce is grown in, Arizona and California. But still, even though we can grow it close to the consumer, can it stay in their fridge and be high quality as long as possible? Appearance or color. So if we're growing a purple leaf cultivar of lettuce, we want it to be purple. Or basil, for example. Basil is a good example because the purple leaf cultivars in general yield much less than the green leaf cultivar. So if you don't care about the color, just grow a higher yielding cultivar. Today, we're mainly going to focus on nutrition. So one of the advantages of controlled environment agriculture is that we have this ability to control the environment, right? That's in the name. But one of the gaps, the biggest gap, is that how do we actually control the environment? So the engineers come and say, okay, what environmental parameters do you want? What is optimal? And optimal is this elusive idea that we're trying to find, but there's so many different factors that go into that. And it depends on, the crop genetics, all these different environmental factors, the cultural factors, and then also what our goal is, right? Whether it's just yield or if it's nutritional quality, and how do we balance all of these together? So the advantage of CA is that we can control the environment, but the big gap is how do we do that optimally? So starting off with, we had a grant through the Tennessee Department of Agriculture. So this is just Tennessee data. Which of the following techniques do you perceive as being the best option to improve micrograin nutrition? And of the consumers, we didn't educate the consumers or provide them any other information. They thought that plant species selection and lighting changes were the best ways to improve micrograin nutrition. So today I'm going to focus on light. So manipulating light intensity, we're going to have this very broad hypothetical idea first. So we have light intensity on our x-axis here, and on the y-axis we have net photosynthesis. You're in a department that folks work really in depth in photosynthesis. We're not going to go really in depth right now. I do have a student working on photosynthesis, but not going to share his data. But okay, we have this hypothetical photosynthetic light response curve, right? And we have our light compensation point, light saturation point that you're familiar with. And maybe that's the curve for photosynthesis, but maybe some of our different compounds of interest have a different response to light intensity. So maybe even after we pass that light saturation point, we can have further increased biosynthesis of some of our compounds of interest. So beyond just photosynthesis and yield, we want to elucidate whether there are other potential benefits, particularly in the compounds that are, potentially induced by stress responses like high light. So some of these that we're working on, anthocyanins, so that those two lettuce pictures, that one with the more purple coloration is from this group of compounds called anthocyanins. Different importance in plants' photoprotection, importance to humans, purple pigmentation, like that's important to humans because sometimes we want that purple leaf cultivar. Potential antioxidant activity, different carotenoids, importance in plants, different light harvesting, so part of that antenna complex, photoprotection, structural stabilization, importance in the human diet, the main dietary source of provitamin A, important in eye health. So some of our NASA work, they're interested in particularly zeaxanthin because they have this issue called SANS, and I can't remember what the acronym stands for, but they have swelling of some of the nerves in the eye. That is a big problem. Different potential antioxidant activity. We're also interested in some other compounds as well, like vitamin K, B and C vitamins. So moving on to kind of the first study. So this study was conducted in a greenhouse, and we focused on daily light intervals, so the total amount of light over the course of the day. Fairly straightforward work. And this was conducted, the actual plant production was conducted at the University of Kentucky in the lab of Dr. Garrett Owen, who's now at Ohio State University. And his undergraduate student, Natalia, grew these plants. with different daily light integrals, and it was fun. I'm sharing this because I'm hoping that maybe we have some collaborations like this between UT and UGA in the future. But I took a van of students up to Kentucky, and the Kentucky and Tennessee students all harvested the plants together. You can see one of my graduate students here measuring photosynthesis. He was generating photosynthetic light response curves. And then we took a bunch of plant samples back to the lab, and my undergraduate student, William, quantified some of the phytonutrients in these lettuce leaves. So it was a really fun collaboration, and students from both universities got to interact and worked on a research project together. So some pictures of the plants. As we increase that daily light integral, that total amount of light over the course of the day, that purple oak leaf lettuce, we had that more purple coloration. And since we did the phytonutrient part, I'll show some of that data that William generated. And what we found was that as our daily light integral increased, the concentration of a lot of these compounds decreased, which is not what we were expecting. And this was with, again, this red oak leaf lettuce. And for the green oak leaf lettuce, we don't have that difference in coloration, but we still have this difference in the head size. But again, we saw some decreases in the concentration of these compounds. So this was over in greenhouse, over the light was applied, these lighting treatments were applied over the entire course of production. So later on, I'll talk about some indoor research with the lighting over the entire course of production, but this isn't exactly what we expected. And we're still delving into some of this data and the photosynthetic responses. But what if instead of applying, say, more light over the entire production duration, we just focus on propagation? So during that propagation phase, that seedling may take up this amount of space, versus when that plant is transplanted into the finished production stage, it can take up this amount of space. So we can fit many more plants in that same space. In this example, and this is just a hypothetical example, we could fit 36 seedlings in that same area. And if we take the production duration of some of our lettuce or potentially basil into account, propagation is about 2 weeks, the finishing stage is about 3 weeks, so 54 seedlings in the same area as one finished plant. So with that, we could potentially spread that cost out across many more plants and have higher inputs during propagation. And if we have higher inputs, can benefits persist through finishing in a common greenhouse environment? One of the main concerns in particularly indoor production right now is the economic viability. And maybe we could have a hybrid between indoor production and greenhouse production, where we have young plants that are off to a really great start. We could potentially spread that greater indoor production cost across many more plants, and could it be viable? So this was, again, a light intensity experiment, light intensity during lettuce seedling production. This is the student Dustin Del Moro who is working on that project. And then he was doing the production piece, and then Spencer Givens and Sarah Armstrong completed the lab portion. And Dustin and Spencer were undergraduate students. So I'm sharing some of my initial research. So I guess what I didn't say at the beginning was I've been at the University of Tennessee 3.5 years now. So yeah, building up the lab. So this is some of the earlier work we did, some with graduate students and a lot with really awesome undergraduate students, which was amazing. But the objectives of this study in particular were to determine the extent seedling light intensity influences yield, carotenoid, chlorophyll, and anthocyanin concentrations. And then would those differences persist through finishing in a common greenhouse environment? So we had 2 different cultivars. Rex is a green leaf butterhead lettuce, and then Theodore is a purple leaf butterhead lettuce. And we had 5 different target light intensities ranging from 60 to 600 micromoles in a 24-hour photo period. Wanted to have very different daily light integrals ranging from 5 to 52 moles. And what we found was that, okay, with Theodore, that purple leaf cultivar, as we increase that light intensity during propagation, fresh mass increased at transplant. And with Rex, we saw similar trends. So we're increasing that light intensity from 60 to 600 micromoles for a 24-hour photo period. we had increases in fresh mass. So some people thought we were crazy. 600 micromoles is really high, particularly for young plants. So what we did was we sowed the seeds and put them right under this high light intensity, and the plants still did really well. Anthocyanins. So we quantified total anthocyanins, and I like this picture along the top here because looking down at the plants, you can tell that this is all the same cultivar. Low light intensity, high light intensity. They don't even look like the same cultivar, right? Very different appearance. And with anthocyanins, you can kind of see that trend as well. So as our light intensity increased from 60 to about 400 micromoles, we had increases in anthocyanins. Then we started to see this little bit of a dip. Now, if we look back at this picture, these plants look much deeper purple than these plants. Like, why do we get this dip? We don't have the data for this project, but my grad student working on this NASA project has some data to back this up. But with that higher light intensity, we're having thicker leaves, and the anthocyanins are in that epidermal layer. And why I'm pointing this out is that sometimes we're using imaging to somewhat quantify, like if we take a top-down picture of our head of lettuce, and we're just using that to say, okay, this one has more anthocyanins than this one. That's more, what is the context of more? Maybe it's a deeper purple coloration, but that leaf is thicker, so the concentration is potentially less. Carotenoids. So what we found from a carotenoid standpoint was with Theodore, we tended to see this kind of increasing quadratic trend. So the most carotenoids, depending on which carotenoid we're looking at, but 200 to 400 micromoles. For Rex has much lower carotenoids in general. And some other folks have found a similar result. But we see, decreasing concentrations with increasing light intensity, and also the somewhat similar quadratic response. So a lot of cultivar differences. So, okay, that's the data when we transplant those plants. And that may be relevant if we're growing potentially baby leaf lettuce or something, even though those are really small plants. But what we're really interested in is, do any benefits persist through finishing in that common greenhouse environment? Fresh mass. So again, the only difference was that propagation light intensity. And what we found was with both Theodore and Rex, increasing that light intensity up to 200 micromoles, maybe 400 for Rex, increased that fresh mass. So beyond that, we did not have a benefit from a fresh mass perspective. But remember, we're also interested in phytonutrients, right? So what happened with phytonutrients? So this was our anthocyanin concentrations at transplant. But what we found at finishing was that there were no differences. So a little bit disappointed, but not surprising. Carotenoids, though, this was surprising. We had the opposite trend. Remember, at transplant with Theodore, we kind of had this increasing quadratic response. And now we're seeing this decreasing quadratic response. And with Rex, lower concentrations in general, but we still see somewhat decreasing trends. So this is something we want to look into more because I think it could be important from a dynamic environmental control perspective. If we look at, so this is concentration, so amount per gram, or maybe think of it as like amount per bite versus content or the amount per head of lettuce. So if we take that yield into account, for Theodore, the amount per head increases with increasing light intensity, and Rex, just much lower content in general, but we saw somewhat increasing quadratic trends. So seedling production versus finishing, I think from a, from a fresh mass standpoint, if we're just looking at transplant, if you're producing transplants or maybe baby leaf lettuce, increasing that light intensity up to about 600 micromoles increases fresh mass, leaf number, and anthocyanin concentrations up to about 400 micromoles, but decreases that Rex carotenoid concentration. But then at harvest, we see increases in leaf number and no difference in anthocyanin, but decreasing carotenoid concentration, so somewhat opposite, but increasing carotenoid content because we're taking that yield into account. So for greatest yield, propagation light intensities of 200 or greater for Theodore or 400 or greater for Rex. But if I were giving recommendations for a grower, I just focus on yield because our trends from a phytonutrient perspective are not really as expected, right? Like we need to be able, one of the advantages of controlled environment agriculture is that we could potentially predict, okay, this is how much we're going to have, and we need to be more confident in the responses and why this is changing. Why are we having these opposite trends at transplant versus at finishing? Which means that we'll have a lot more research questions over my career, right? So this study here, one of my graduate students, Ethan, who I'll share his data later, he was working completely indoors, and he said, Dr. Walters, I want to work in the greenhouse. But he had so much on his plate. I said, how about you mentor undergraduate students in the greenhouse? So he was working on green Amara, mustard, and hybrid red Mizuna. And he grew these seedlings, again, under different light intensities, 200 to 800 micromoles for a 16-hour photo period. And Lauren here did the production piece. So she took those seedlings that were grown indoors and grew them out in the greenhouse. And Maggie here did the phytonutrient quantification. So what we found was, okay, now we're not just growing lettuce, but we're growing these Mizuna and mustards, that at transplant, as we're increasing that light intensity, we have increases in fresh mass, which is what we expect. And then we grow it out in the greenhouse in raft hydroponic systems. And what we found was that these increases persisted through harvest. So growing that Mizuna or mustards at up to 800 micromoles of light for a 16-hour photo period actually had benefits in yield that persisted, which was awesome. So what's going on behind the scenes, what we can't see? I just pulled out a couple of examples. So per gram concentration per plant here. And these are some B vitamins. So B3, B6, B7. And what we see, and this is all at transplant, so not at finishing, just at transplant, as we increase that light intensity from 200 to 600 micromoles, we're seeing increases in vitamin B3. We tend to see some increases in B6 and B7 as well. The green bar is green Amara, and kind of the red one is hybrid red Mizuna. If we take yield into account, we kind of see these same trends that kind of persist. But if we transplant them into a common greenhouse environment, even though we had differences in fresh mass that persisted, we had no differences in any of the phytonutrients, not just these B vitamins that persisted. So very species and cultivar dependent. Okay, so you probably can't see this. This is a moody picture of some deep purple basil, but I'm not going to talk about basil. I just really like the picture. But should you germinate in the dark? So we're doing these studies and we're sowing the seeds and sticking them right under these lights. And talking too much, I'm not going to get to the cool NASA stuff, but people are like, should you germinate in the dark? So we designed a study to test this out. And Dustin, who grew the plants for that first lettuce light intensity study, also did this. So we have days covered here on the x-axis, and we have these two bars with 200 and 800 micromoles. So low light and high light. And we go from, and this is, well, I guess I am talking about basil. We had the same trends with lettuce and kale too. So these plants were put straight underneath the lights, covered for 1 day, 2 day, etc. So the plants that were put straight under the light or covered for 2 days had similar fresh mass at transplant. If those seedlings were covered for 3 or more days, we started to see a decrease in that fresh mass at transplant. For the plants that were grown under that high light intensity, but no difference in the plants that were grown under the low light intensity. At harvest, what we saw, again, that low light intensity, no difference in that fresh mass, but we started to see some decreases in that yield that persisted. Also, the plants ended up being having, oh, never mind, I have this slide. Okay, at transplant, we have hypocotyl length. So kind of how stretchy are the plants, right? So, or how leggy, but they were covered for 2 days, very similar hypocotyl length. They were covered for longer. We started seeing more stretching. It was also very difficult to transplant and get the seedlings apart because they were all starting to intertwine. And at harvest, we saw some of those differences that persisted. So for basil, lettuce, so this is Italian largely basil, Rex lettuce, and can't remember, some kale. Cover them for 2 days without any decreases in yield or hypocotyl length that persists. So, okay, that's the beginning of production. What about the end of production? Can we do something quick to improve the phytonutrients? We don't have that time, in a common greenhouse environment for some of those benefits to get washed away. But what can we do? So again, we're focusing on light intensity right now. So my former graduate student, Jessica, worked on this, and then a bunch of folks helping out. So these were her deep water culture systems in the greenhouse. Here are the plants growing. So she grew red borer and winter borer kale, so a red leaf cultivar, green leaf cultivar. And this was in a greenhouse propagated in ebb and flood systems, and then grown in deep water culture systems. So we conducted this in the greenhouse, and then we also conducted this indoors. So the same kind of air temperature of about 22 degrees, and our target daily light integral was actually around 10 moles of light indoors, so 175 micromoles for that 16-hour photo period. And today I'm mainly going to share the greenhouse data. So end of production, what is end of production? Is it 1 day, 3 days, 7 days? And then we had different light intensities. So we're adding on top of those 10 or 11 moles of light, 100 white light, which is what I'm going to show today, red and blue, 200 micromoles of white light, 400 or 600 micromoles. And what we found was that, okay, as we increase the light intensity for a longer duration, we have increases in leaf number. We have increases in fresh mass that we would expect, right? Because we're just adding more light. But what our goal was to increase the quality, the nutritional quality of these crops. So anthocyanins, we have our winter borer here, which is a green leaf cultivar, so not many anthocyanins. With red borer here, we tended to see some increases in anthocyanins with that highest light intensity for either 3 or 7 days, or even 600 or 400 or 600 micromoles. So what did these plants actually look like? So this was our control plants of red borer, which you can hardly tell they're a red leaf cultivar. 100 micromoles of red and blue, a little bit more pigmentation, 100 micromoles of white, 200 of white, 400 micromoles of white, and 600 micromoles of white. So you can see that as we move from this area up here, those leaves become more purple. Now, what we found indoors, I don't have slides to show this data, but in general, it took a lower light intensity and less time to achieve greater pigmentation. And this kind of makes sense, right? In the greenhouse, we have more variable light, variable temperature over the course of the day. So when we increase that light intensity, it's not as stressful, right? The plant doesn't need as much to adapt to that greater light intensity versus indoors, we have 175 micromoles. And then all of a sudden we increase that, we have a much stronger response. And with the high light intensities, we even had some burning on the leaves. So furthering some of the data indoors, some of the water soluble vitamins. So Kathleen Kaufman, who's working on her PhD at Oak Ridge National Laboratory now, she did this quantification, but depending on the treatment, increasing our additional light at the end of production tended to increase some of the B vitamin concentrations. Now, something else I want to point out is this. So this was done by, done by Kathleen, but these trends are opposite, right? So we have red borer and winter borer kale. Most of the water soluble vitamins, the B and C vitamins are vitamin C that was there. cultivars respond differently? And why do they respond differently? So these are some questions that we need to investigate if we want to have predictable phytonutrient responses, right? So just more questions. At first we look at this and it's like, oh, dang it. We just want a straightforward answer, but it's not straightforward. So end of production lighting, good strategy for coloration. More information is needed for consistent phytonutrients. Okay, so what do consumers think? So this, going back to that initial survey that I showed you, we did this study on microgreens. So we had two different brassica cultivars, or species, kale and radish. And then we had two different cultivars of each, a red leaf cultivar and a green leaf cultivar. And then two different light intensities, 175 micromoles, 575 micromoles, so high and low. This is what the plants looked like. So the goal for this study wasn't to do groundbreaking plant physiology research. It was to create differences and assess what consumers think. So brassicas have this class of compounds called glucosinolates that are anti-carcinogenic. They're good for you, but they're not pigmented. Anthocyanins impart that purple coloration. So do consumers have different conceptions or preferences for different beneficial compounds that you can see differences versus things that you can't see? From a anthocyanin concentration standpoint, here are red cultivars. You can see the axis is much different between the green cultivars. But usually we think, okay, we're increasing that light intensity. We're going to have more anthocyanins or a greater anthocyanin concentration. But that's not what we found in that red rainbow radish. We actually saw that decreasing concentration. But if we move on to height, the height was greater with that lower light intensity. So we had more of that elongation, which can be potentially beneficial for microgreens. And actually consumers did prefer plants with that longer hypocotyl. Whole plant dry mass. So again, that higher light intensity, even though that lower light intensity, they were taller, but the plants with that higher light intensity have more mass. So some of the consumer data. So Tennesseans' preferences for microgreens of a different color. So we'll look at these two here in the middle, green kale and green radish, compared to light purple kale and radish. There was a price premium of 52 to 66 cents. Now, if we compare dark purple kale to light purple kale, there was a price penalty of about 50 cents. So Tennesseans do not like purple leaf cultivars, which surprised us. But I talked to someone in Arkansas and they said they had similar differences. So it's like, as researchers, you know, we think one thing, but the consumers may think something else. Also, okay, which of the following techniques do you perceive as being the best option to improve microgreen nutrition? So this is the graph we showed earlier. Lighting changes and plant species selection was the best, or none, but the best of the different changes we could have. And if phytonutrients were enhanced by light, the consumers were willing to pay 47 to 79 cents more. So just saying that it was enhanced with light, so no additional information. I thought this was interesting too. If you were seeking nutritionally dense microgreens, how safe for consumption would you perceive the following methods to enhance nutrition? And the safest was lighting changes and plant species selection. So what people thought were riskier strategies to improve nutrition, they decreased their willingness to pay, which makes sense. So they think gene editing and genetic modification is more risky, or even using fertilizer is risky. So controlled environment agriculture, great challenges, but great opportunities. So now I want to share a little bit of, kind of separated this out because we have a nice story here. This is all the work of Ethan Darby, who just graduated. Right brassica carinata for space production, cut and come again protocols for yield and phytonutrient optimization. So I'll be sharing a little bit of his thesis work, and this was actually the first grant proposal I submitted as a new faculty member with an 85% research appointment. I started about 2 weeks after I graduated with my PhD. And I'm panicking, I'm like, oh my gosh, I need to get grants. So I think, okay, if I could do anything, like all the blinders off. So take all your blinders off when you're thinking about it. What would I want to do? I would want to grow plants for space. So I submitted this proposal, my first federal proposal, and it was fun. So just if you're passionate about something, I think it comes through sometimes. So this was Ethan's work, and even though I'm passionate about this, he actually did all the work. So what are the space issues? So the diet is deficient in certain nutrients. The stored space diet, so that freeze-dried diet, is deficient in vitamin K, for example. So if you just ate that stored space diet, you might become deficient in vitamin K. Health risks of radiation and microgravity. So I mentioned before that eye swelling and how zeaxanthin might help mitigate some of that effect of that eye swelling, particularly in male astronauts. And yeah, radiation and microgravity in general has a lot of negative effects on the human body. And the plants can produce some compounds that can help mitigate that. Degradation of compounds during long-term space flight. So, okay, you decide that you're going to be an astronaut, you're going to go to Mars. Now, if he's only eating that stored space diet, some of the compounds are degrading over time, like vitamin B1, vitamin C. You don't want to get scurvy. We thought we were over this problem, but luckily plants can create vitamin C. So we don't have to worry as much about the degradation if we can just eat these plants. Fresh produce as a solution. Again, actively synthesize these sensitive vitamins. No need for storage. You could harvest them and eat them if everything is timed right. Provide synergistic benefits and helps avoid potential overaccumulation. So potential synergistic benefits with other compounds that are in plants or potential antagonisms. And overaccumulation, if you're like me and take gummy vitamins because they're just really tasty, you may take too many of them. Anyway, they're concerned about that. Mizuna was identified as a promising candidate due to their nutritional and flavor characteristics, and also their phytonutrient profile. So that's how we chose Mizuna. And some of the parameters of interest. So, okay, we're growing Mizuna. We have these different problems. Plants can create things, but what are we actually interested in? Water-soluble vitamins, some of what we mentioned earlier. So for this study, we focused on vitamin B1 and vitamin C. Lipid-soluble vitamins like vitamin K. Minerals, we're particularly interested in calcium, potassium, and magnesium, and iron. Then carotenoids like zeaxanthin that we talked about earlier. There's also provitamin A. Anthocyanins as potential antioxidants. And then morphology. So we're interested in morphology. We want something to grow fast, so great fresh mass, but space is a constraining factor in space. So we also want something that is compact. So mass in a compact package that is filled with everything that we want, right? So that's the goal. So dietary needs and space-based limitations. So, okay, we're interested in all these parameters, but how we're evaluating these is based on this weighting scale. And this was published by, I should have a reference, Joy Amasa. She's a plant scientist at NASA. And we have positive weighing for things that we want, like fresh mass, but plant volume, we want that compact plant. So that's negatively weighed. So you'll see the importance of all of these based on their weighting factor. Some things are negative, like iron, which you'd think, oh, I thought we want more iron in the diet, but actually increase in iron in space is associated with an increased rate of bone loss. So for these studies, we had some different environmental parameters. So we tried to emulate, this was all at the University of Tennessee, all on the ground, much less expensive than actually growing on the space station for these large studies, especially. But we had a target temperature and relative humidity similar to what is on the space station. So we didn't have to adjust that. And then the carbon dioxide concentration was about 2,800 parts per million. So really high, higher than we would have here on Earth normally, around 400 parts per million. But it's important to have that when we're doing this work because Mizuna was actually the second highest rated leafy green that they evaluated. The first was Chinese cabbage. They did all this ground-based work, and then they did some research on the space station, and they had chlorotic and epinastic growth. I'm like, what the heck happened? Come back to Earth, do some more follow-up studies, and it was carbon dioxide concentration, because their ground-based studies were concentrations like 1,000 to 1,500 parts per million, but that super high concentration yielded some unexpected results. So that's why we kind of had that environment. We used deep water culture hydroponic systems because they're having a lot of watering issues on the space station due to the lack of gravity. They've been using these plant pillows. They've been experimenting with different methods of watering, but they're not satisfied with any of them. And they're all having different drawbacks. So that's something that a lot of folks are working on right now. So they said, use whatever system you want, deep water culture. So our first study was a cultivar evaluation. And so we had about 22 different cultivars of Mizuna, Mibuna, Brassica carnata. It was like this Mizuna Asian green continuum. I just ordered all the seeds. And they're all, you can't see all of these pictures, but they all look quite different. And it was fun to see, but they all had very different growth rates. We don't have all 22 cultivars here because some of them were too small to even get all of the data. But they had different phytonutrient profiles as well. So we weighed all of these, normalized the data, weighed them based on that scale that you saw earlier. And our top cultivar, again, based on that scale, was Green Amara. And the other cultivar that we decided to go forward with was hybrid red Mizuna. Now, Green Amara has higher vitamin B1, for example, but it has a lot of iron. So we chose Green Amara and hybrid red Mizuna because some of these different phytonutrients were complementary. So you could potentially grow them both based on what phytonutrients we're interested in. But also they looked quite different. So Green Amara here is green. Hybrid red Mizuna is red. Different appearance and leaf texture morphology. They also taste quite different. Hybrid red Mizuna can be spicy versus Green Amara. I described as a little bit more meaty, as meaty as you can get for a green, I suppose. But yeah, balancing some of this. So today I'll be focusing on Green Amara here. And the next step, taking these two cultivars forward to the next study, a light intensity photoperiod interaction study. So we had 4 light intensities ranging from 200 to 800 micromoles. And we had 2 photoperiod, a 16-hour photoperiod, kind of our normal, and then a 24-hour photoperiod. And our thought was, let me see, our thought was if we can spread that light over that 24-hour period, maybe the plants would use it more efficiently, maybe it would be more energy efficient. Yeah, so we had 9 plants per combination, and we repeated this twice in time. Here are some pictures of those Green Amara plants. And what we found is for that 16-hour photoperiod, as we increased that light intensity, the fresh mass increased. The size of those plants increased. For the 24-hour photoperiod, we saw increases up to about 600 micromoles and then a decrease in mass as we further increased the intensity up to 800 micromoles. If we take all of those different factors into consideration and apply our weighting scale again, what we found was that the highest ranked treatment was Green Amara, grown under 800 micromoles for a 16-hour photoperiod. And then we didn't have all the mineral data in at the time this graph was made, but we also proceeded with the 200 micromole treatment because after we add the minerals, it was quite high ranking, and 200 micromoles is much less energy than 800 micromoles. So then we go to the final study, and this is cut and come again protocols for yield and phytonutrient optimization. So what do we mean by that? Oh, well, okay, first, questions. How does successive harvest affect phytonutrients and biomass over time? So we're growing the plant and we're continuing to cut it. Are our harvests consistent? we're not just worried about yield, but what about the phytonutrients? Does allowing plants to develop more between harvests increase plant productivity over time? So at first we were just going to do the successive harvest experiment. Then it's like, okay, what is successive harvest? Like, should we actually do this? So this study focused on a couple different harvest cues. And the whole thought behind successive harvest is, okay, we sowed a seed, it starts to grow. The growth rate is pretty slow, right? As you know, the plant is just emerging, you have the cotyledons, but as the plant's starting to unfold leaves, that growth rate increases until we start to reach that reproductive phase when growth kind of stalls. So the idea behind cut and come again production is staying in this linear phase of growth as long as possible. Now on Earth, would that be feasible? Maybe, maybe not. There's different considerations for pathogens, etc. But for space, we have a unique growing environment, so we can do all sorts of things. So the experimental design for this was, we had two different kind of successive harvest experiments. The first was a time-based harvest. So we harvested every 10 days. The second was a developmental-based harvest. So after the plant had 8 leaves. Factorial design, 2 light intensities, and then 5 harvests over time. So again, 200 and 800 micromoles, 5 harvests over time. We only achieved the 5th harvest on the time-based experiment when we harvested every 10 days. The developmental-based experiment, the plants, we were waiting and waiting for that 8th harvest, but the plants started to bolt, so it wasn't even feasible. So we only got 4 harvests. Data collection and methods, again, are 2 different, developmental, that every 10 days or every 8 leaves, 2 different light intensities, 5 consecutive harvests per plant. We had 8 samples per harvest per treatment. This was all conducted a couple of times in time. Here we're going to just focus on water-soluble vitamins, carotenoids, and anthocyanins, along with yield. Ethan creates really nice figures. So time-based harvest. So harvesting every 10 days. We're going to share those results first. So this was dry matter percentage. So how much of the plant is dry mass versus how much is water? And so going from the 1st to the 5th harvest, more and more of that fresh mass was dry matter. More results. So that was the main effect of harvest. The main effect of light intensity. So dry matter percentage, and then we have vitamin C, vitamin B1, and lutein. But dry matter percentage and vitamin C and B1 increased. We had greater amount concentration with that higher light intensity, and the opposite was true for lutein, kind of what we saw earlier with higher light intensities, lower carotenoid concentrations. Fresh mass, we had interactions between light intensity and that harvest. So what we saw was that, okay, the dark blue bars are the high light intensity, light blue is low light intensity. And with that high light intensity, fresh mass was greater those first 3 harvests, and then we started to see that dip. Less of a difference with the low light intensity. For beta carotene, then that high light intensity, we had, again, lower beta carotene carotenoid concentrations than the high light intensity, but more variability across harvest with that low light intensity. Anthocyanins, as we increased, as we went through our harvest, we had greater concentrations of anthocyanins. And then zeaxanthin, we didn't have any kind of trends to note. It was interesting with hybrid bred Mizuna, we had kind of no differences in zeaxanthin, but then when we got to the last harvest, the plants that were grown under 800 micromoles, we had a lot of zeaxanthin. Developmental based harvest, so harvesting after every 8 leaves. So this is the main effect of harvest. And what we found was with these 3 different carotenoids, we saw this dip in the 2nd harvest and then an increase in the concentrations after that, which was interesting. Something we want to look into a little bit more. Why did this actually happen? Main effect of light intensity. So again, these are these 3 same carotenoids. the same trend as kind of saw in the studies shown here and in other studies that we have done, that higher light intensity, lower carotenoid concentration. Interaction of light intensity and harvest per fresh mass, we had greater fresh mass, and with that, greater light intensity. Dry matter percentage, a somewhat similar trend as the time-based harvest, that as we go from the first to the fourth harvest, we tend to see greater portion of that fresh mass is biomass and not water. The vitamin C, that low light intensity, those first 3 harvests, we had more vitamin C. So, okay, that's a lot of different graphs I just showed you, and you're probably all starting to glaze over. It's like, what does this actually mean, and how do we actually put all of this together? We just want to grow the plant. So, per diem output. So the goal is, okay, we want to provide you with your recommended daily value every day. So how much are we producing per day, and when should we restart that plant? Or restart that plant, sow a new seed, right? So this is taking into account our light intensities and our 2 harvest methods. So what we see here is these dark blue circles are a high light intensity, or dark blue symbols. Sometimes the words are hard to come by. But the circles here are the developmental based harvest. So it's good that we ask that question, you know, what should our harvest queue be? Because that developmental based harvest had a much higher per diem or per day output of fresh mass than the other treatments. If we look at some of these different phytonutrients, we see a somewhat similar trend, right? Towards the top, we see those dark blue circles. So in general, that high light intensity with a harvesting every 8 leaves has our greatest per day output. Now, if we look at the energy for the lighting required, so yeah, we want to be efficient with our space, right? Because that's a really precious resource. But then we look at energy from lighting, and we see a different trend. We see towards the top here, we have light blue circles, so still harvesting every 8 leaves, but that low light intensity was more efficient from a lighting energy standpoint. Now, this doesn't take into account the energy for, you know, the whole production system or anything else. But I like showing these 2 different figures because we have to weigh all of these different factors when we're figuring out what we should grow and how we should grow it. So we just submitted a proposal. Keep your fingers crossed for a figures of merit analyses on these, all of the inputs and outputs. The goal is to figure out exactly what we need to provide a 40-year-old female with their recommended daily value of these different phytonutrients. So some conclusions for this, that Green Amara provided kind of the best overall platform for that weighting scale that we had, that we used. We're also working on kind of revising that scale because it's just based on the importance, but not based on someone's recommended daily value intake. So for example, one of the minerals might be weighed times 1.5 and vitamin K might be times 1.5, but for one serving of that Green Amara, you might be eating 200% of your recommended daily value of vitamin K, but only 3 to 5% of that mineral. And that vitamin K should be weighed more heavily then because it contributes more to your recommended daily value. So we're working on a new system for that. 800 micromoles, high light intensity applied for 16-hour photo period had kind of the best overall results for Amara. The efficiency generally for successive harvests, efficiency was gained across all metrics with consecutive harvests. So harvesting for that every 8 leaves 4 times was good. Developmental harvest had the greatest output, greatest per day output was with that high light intensity. Greatest per unit lighting energy output was with the low light intensity. So I know I always talk a little bit too long. I wanted to talk about a couple of the projects that we have going on right now. So we don't just work with light intensity, we're doing some work with mineral nutrition. and other lighting factors. But this is one project that we have. I led a 6 PI team, and we're focusing on different end of production strategies. Can we do something quickly to increase the quality of the crop, whether that's phytonutrients, appearance? We have a post-harvest longevity component. So different lighting strategies, temperature, nutrient solution strategies. And then we have economic feasibility and consumer preference, and then different training resources we'll be putting out. I also co-led a big project called CEA Herb. So we're doing a lot of culinary herb work. There's 3 main kind of aspects: marketing, economics, production, et cetera, and engaging stakeholders. And my PhD and master's was working with culinary herbs, and I'm excited with this because we're deploying a lot of different production strategies to improve efficiency, but we're also doing some metabolomic work with the volatile compounds and what exactly changes and how can we have the best quality culinary herb as well. Everything from that to pairing with consumer sensory perceptions and willingness to pay. And my collaborator has these eye trackers with all the marketing stuff, which I think is cool. But humans are weird, so I'm glad we just work with plants. Some acknowledgements of funding and things. And then I have a lot of folks in the lab that have helped with some of the studies I shared here. And these aren't all current students. These are all the students who have been in my lab in the last 3.5 years, students and postdocs, et cetera. They're the ones that did the actual work. Any questions? Yeah.
Could you grow a healthy lettuce for the NASA study by reducing iron in your nutrient solution so that the plants don't accumulate as much iron?
So could you grow a healthy lettuce plant by just reducing the amount of iron in your nutrient solution? Yeah, you could reduce it to a certain extent, but it depends on the plant and how much it takes up. And the plant still needs iron to grow, and some plants need more than others. Something that I'm working on with a collaborator is iron bioavailability. So our goal in our initial conversations were, how can we make, say, iron in spinach more bioavailable? Because the spinach has a lot of iron, but the bioavailability is actually quite low. And why is that? And again, our goal for Earth is to increase the bioavailability, but we can also use that to, even though the plant has iron that it needs to grow, et cetera, but reduce the bioavailability for astronauts. Other questions? Yeah.
So when you look for the phytonutrients, did you also look for the phytotoxins? Or you specifically going to look for the nutrients which is beneficial?
Yeah, so when we examine these phytonutrients, did we also examine different toxic compounds that may be in the plant? No, we didn't. We just targeted these phytonutrients. Yeah.
I had a question. When you did your consumer surveys, did you educate those consumers on all of those factors that you took? Like, what does gene editing do? What does lighting do?
We did not educate the consumers. So we wanted just to see what normal consumers would say. And that's what one of the reviewers was like, why didn't you educate the consumers? And it's like, we're not going to educate all of the consumers all the time. So we just want to know what normal Tennessee folks think. Yeah.
Where were those surveys conducted?
So some of those surveys were conducted online, and there was about 900 people who participated. And then we also did in-person experimental auctions, where I grew a bunch of microgreens. And then I didn't actually go to the auction because I couldn't make it that day. We need another one that I can go to and see, because it sounds really cool, but have the microgreens in a clamshell, and the consumer actually bids real money on how much they would pay for it, based on telling them, okay, this one is more nutrient dense, and there was a piece having them taste it, and yeah.
Did you tell them one of them was gene edited? Or?
No, no. The experimental auction mainly focused on the nutritional quality aspect, and just saying, okay, this is more nutritious, and because that was my main goal, because we're doing all this phytonutrient work, but do consumers care? Yeah, even if there's no increased willingness to pay, my goal is to provide tasty, nutritious food to everyone at an affordable price, improve access. But yeah. Still, are consumers willing to pay more for this to continue improving some of this quality?
Were there any questions about, like leaf thickness or morphology for the consumers?
Yeah, were there any questions about leaf thickness or morphology for the consumers? There was a question about not leaf thickness, but plant height. And the consumers preferred the microgreens that were taller. So they preferred the ones that were grown under that low light because of the stem length. Yeah.
Yeah, quick question on the survey. So I saw when you asked like some factors that can help improve the quality, and many of them say none. Like, was there a follow-up question? Like, do you know the mindset behind that? Like none.
I did that in conjunction. I did the survey in conjunction with Alicia Reed. She's an economist. Who's a wonderful human being. So I don't remember all the survey questions, but I don't think there was a follow-up question to why they selected none. But yeah, it's interesting to see what folks think, like fertilizer, like we have to fertilize the plants in the field, like that's kind of a normal thing. But consumers think that's somewhat risky, and maybe we just, because we use the word fertilizer, and I think there's a lot more questions that unpack there. And again, this survey was only in Tennessee. It'd be interesting to see what other parts of the country, how they perceived things, because some of these results were across, we thought, okay, maybe it's just like rural areas, not like Nashville. But we saw some of these same results in Nashville. So it's not just this rural area, not used to the purple leaf lettuce and yeah.
Or even microgreens.
Or even microgreens in general, yeah.
So Kelly, you did research with grown lettuce, for space travel and living. So how about using microgreens that occupy way less space, to produce? Because I was thinking the size of the production units that you would have to have in space to feed a proportion of the diet when fresh. So
Yeah, so should we just use microgreens? Microgreens are nutrient dense, right? And actually, Joya Massa at NASA is conducting a study with the USDA on microgreens and microgreen nutrition. Now, all of the work I'm doing right here is I grow the plant, harvest the plant, and we quantify the phytonutrients. But just kind of like with that iron, we're not taking into account the bioavailability. So my collaborator, Mike Jazakovich, he's joint appointment with Baylor and the USDA and Baylor Medical Center and the USDA. He does really cool work with bioavailability. And I sent him some of these microgreens that we grew. And he was looking at carotenoid bioavailability. And I also sent him some of Ethan's plants too in a failed run, but it wasn't failed because we got really excellent bioavailability data. But the microgreens, he looked at the data and then ran it again because the bioavailability of the carotenoids was really low. So look through the literature, someone else had reported this as well. We're hypothesizing that there might be some interaction with fiber or something else that is causing that reduction in bioavailability, particularly of carotenoids. We didn't assess any of the other compounds, but yeah, so maybe microgreens aren't as nutritious as we think. Now, that might not be true for like vitamin C or the B vitamins, but yeah. Other questions?
You
Yeah.
mentioned that you measured the anthocyanins in a non-destructive way. But did you try with other phytonutrients? Same
So
strategy.
we didn't actually measure anthocyanins in a non-destructive way.
Mhm.
So everything that we did was destructive.
Okay. Did you see potential on like going into a non-invasive way of, or did you try and compare with what you got destructively?
So we do, Julia, one of the undergrads in my lab, is taking some of our image data and doing color analyses, particularly to compare with our anthocyanin results, because we quantified anthocyanins on all of these and we have all these pictures. So I just said, have at it with the data. So she's working on that. We have a different collaboration with Stephen Lanton at the University of Florida. He's in engineering. He works with hyperspectral imaging. And he had some NASA funding with using hyperspectral imaging to quantify beta carotene and lutein because they're pigmented compounds. So maybe we could use these as a strategy. So we're doing the lab analyses for him. And yeah, so to be determined. We're in the thick of it. Yeah. Any other questions? One last thing, if you guys are ever in Knoxville, don't hesitate to say hello and stop by. You're welcome. Thank you.